Hi guys, welcome to my presentation for today. Today we're going to be talking about the effects of transcranial direct current stimulation and its impacts on physical exercise. So let's get started. So what is transcranial direct current stimulation, or TDCS as referred to from here on out? TDCS is a method of non-invasive brain stimulation that results in the excitation of the brain due to the application of a weak electrical direct current, as from Kamali et al. 2019. TDCS alters the action potential threshold in neurons. Now there's two types of TDCS. We've got anodal, which is excitatory, which causes depolarization of the neuron by increasing neuronal firing rates and cortical excitability. And then we have cathedral or inhibitory TDCS, which decreases excitability through hyperpolarization, as found by Nitsch and Paulus in 2000. Furthermore, tDCS transmits a weak current of 1 to 2 mA through surface electrodes placed over the scalp for a duration of between 5 to 20 minutes, as found by Angius, Hopka, and Morga 2017. This electrical current transfers through the brain tissues and influences the excitability of neuron and thus neural activity. Research by Nitsch et al. 2008 found that 10 minutes of brain stimulation had effects that lasted up to 1 hour after the intervention. Furthermore, through the use of functional magnetic resonance imaging studies, or fMRI studies, TDCS-induced stimulation was found to influence blood oxygen levels in the targeted areas. Now, this review is going to investigate the impacts of TDCS on physical performance, which will be defined as any bodily movement produced by skeletal muscles that require energy expenditure, according to the World Health Organization in 2014. Now, Moscatelli in 2016 found that excellence in physical performances requires not only physical but also neural motor capabilities and sensory cognitive skills, which highlights the interconnected nature of brain structures and functions. Going a bit more specific, however, and anatomically, the primary motor cortex, or M1, is a subsection of the brain which plays a pivotal role in motor performance and motor learning due to its interconnection with the parietal lobe, premotor cortex, supplementary motor area, basal ganglia, and cerebellum, which was found by Keyshaw 2014. And furthermore, Keyshaw 2014 found that the M1 had the capacity to offer behavioral flexibility to motor functions, which was dependent on its ability to modify its local neuron architecture and promote dynamic neural motor maps. The M1 is going to be the focus of this investigation due to its vital role in physical performance and motor neuron coordination. Now, as society is becoming increasingly driven to succeed and perform at higher levels for longer durations, performance enhancement strategies such as TDCS have gained widespread awareness. Now, in order to increase physical performance, fatigue should be minimized, as it often reduces our ability to perform physical tasks for extended periods of time. Now, supraspinal fatigue is a failure to generate output from the primary motor cortex, or M1, which contributes to the development of muscle fatigue, as found by Gandevia in 2001. Further studies by Taylor et al. 1996 found that supraspinal fatigue is correlated with changes in motor cortex excitability. Thus, interventions that could increase M1 excitability are hypothesized to increase M1 output, thus delaying the development of supraspinal fatigue, therefore improving physical performance. And this was found by Cogman et al. 2007, Williams et al. 2013. And now this is where TDCS has an opportunity to excel and modulate that excitability of a targeted brain region such as the M1. Now, exactly how TDCS will improve physical performance is still unknown and unclear. However, it is suggested that TDCS facilitates the M1 by increasing its output during exercise by reducing supraspinal fatigue, found by Angus 2017, Cogman 2007, and Williams et al. In 2013. It should also be noted that anodal TDCS was used to reduce the release of inhibitory neurotransmitter GABA, which contributes to the feelings of fatigue and muscle exhaustion, which was found by Stagg in 2019. However, it is important to note also that it's quite inconclusive to see exactly how TDCS influences physical performance, and further research is certainly needed in the field to see conclusively how it occurs. But we know that there's a strong body of research that suggests something does occur, but further research is needed to conclusively determine how and more specific what mechanisms are involved. And before we get started, I just wanted to mention that Williams et al. 2013 used annual TDCS during physical activity, elbow flexions, and found there was a correlation between TDCS and increased performance. They found that time taken to fatigue the muscle group had increased by more than 30% and prolonged the period of time that the subjects could sustain a high degree of effort. Now, as we move on to current research critiques, there's a few inconsistencies that are explained by the inter-individual variability. And this is highlighted by O'Connor et al. Study in 2015, who targeted the temporal cortex, and he found that through TDCS, he was able to enhance maximal power output by 4%. However, using a similar methodology at a later date, over the same brain region, they found no effect on cycling physical performance, which was measured by maximal output using the same methods of tDCS, which is quite interesting to see that kind of inconsistency. That may be explained by the electrode montage and location. However, further research is definitely needed, and it's quite common to see this inconsistency across the literature on the relation between tDCS and physical performance. So that definitely needs to be addressed. Now, as we move on to future research suggestions, research is actually quite limited on the effectiveness of tDCS on physical performance due to flawed methodological designs and a wide range of extraneous biological and psychosocial factors. And now, while tDCS seems promising, further research is required outside of the laboratory setting that account for the biopsychosocial factors such as existing health levels, long-term effectiveness, and structural changes to the brain. However, being said, the body of current evidence suggests that tDCS has a positive effect on physical performance. Further research is also needed to conclusively determine if tDCS is effective in improving physical performance in elite athletes, as it's commonly only used in kind of your average person, such as uni students and younger people. It's important to know that tDCS has the potential to enhance motor learning and should be investigated to improve other cognitive functions. As it influences motor neuron excitability, it may have a case in offsetting the natural effects of aging and neural decline. So therefore, that's quite an important field that that research probably could get into. That is all we have for today. There's just a few references over here. So the next few are just a couple of references. If you have any questions, please don't hesitate to let me know. Thank you very much.